Hello, freshmen, honors biology. We are chapter 21, going through viruses and prokaryotes, and this is, yes, the good, the bad, and the ugly. Good morning. All right, so we previously talked a little bit about viruses and where they fit on taxonomic scale, and they don't fit anywhere. If you think of the tree of life, and that tree is branching out, one branch is the prokaryotes, which can be divided into the extremophiles, the archaeobacteria, as well as the typical bacteria, and then you have eukarya. So if you have those main branches going out, maybe you could think of viruses as vines that are wrapping in and out and around of all of those living organisms. So yes, bacteria are susceptible to viruses. Eukaryotes, like ourselves, and plants are susceptible to viruses. And here we go. This is the good, the bad, and the ugly of viruses, and bacteria for that matter. Notice here we have viruses. These are non-living, as we talked about in the previous video. So they do contain DNA or RNA, but they are not cellular. They don't have a metabolism. They cannot reproduce on their own. They can, however, reproduce when they get inside of a living cell. So let's go ahead and get this. We've got a non-living particle made of proteins and nucleic acids. And notice the viruses will have genetic material. That genetic material could be DNA or it could be RNA. It just depends on the specific virus. The protein coat which surrounds and protects that genetic material, that protein coat is called the capsid. So a virus, the simplest virus, is going to have DNA and RNA, DNA or RNA, and have a protective protein coat. Of course, that's a simple virus. Some viruses are much more complex than that. Some viruses they bring in little toolkits when they infect a cell. So they make their way inside of the cell and they bring in their own specialized toolkit to make it easier to take over and control that cell and direct that cell into start doing viral things instead of cellular things. Again, it bears repeating. Viruses can only reproduce by infecting living cells. Make a note that viruses are very, very specific. So a virus will typically infect only one specific type of cell. So the hepatitis virus will infect only liver cells. It won't infect nerve cells. It will not be able to infect liver cells, for example. Why is that? Well, that virus is specifically adapted to going in and attaching to a specific cellular receptor, which is only found on that specific type of cell. So a liver cell has a specific receptor that other body cells don't have. HIV, which we're going to be talking about quite a bit actually. HIV will attach to a immune cell and attach to a specific receptor called a CD4 receptor. Other cells, most other cells don't have that CD4 receptor. This specific type of immune cell, they're called T lymphocytes. Again, immune cells, so they're going to help fight off disease. And as we talk about viruses and bacteria, we are going to talk about how we fight these sort of things, how our bodies produce antibodies to help deactivate viruses and bacteria and other pathogenic organisms. Well, let's take a look at a couple different viruses here. So this is a bacteriophage or bacterial phage. Phage simply means virus. Really interesting shape. It looks like some sort of lunar space module. And what I want you to do is make a drawing of this bacterial phage. Underline phage and then write virus. And then list the parts of the bacterial phage. So notice we have the capsid head. So we have the capsid head here. Well protected in this capsid, which is a protein, is the genetic material. We have these little tail fibers which will help attach this bacterial phage to the unsuspecting bacterium. Notice we have the sheath and the rod. Here we have the flu virus, which is related to the COVID-19 or the coronavirus. And inside we have the genetic material, and then we have the outer capsid. I want you to make a note that viruses are cellular parasites. A parasite is going to attack, infect, and maybe ultimately destroy its host cell. Well, notice, let's go through the points first, and then what I want you to do is copy it down. It doesn't do a whole lot of good to just start writing. I want you to listen here as I go through the points, and then we can copy it down. So notice, a virus is entering living cells. It's going to use the machinery of the cell and produce more viruses. So what is the machinery of the cell? The machinery of the cell are the ribosomes. Ribosomes are protein factories. What else is considered the machinery? Well, for the virus to reproduce and make more viruses, we need energy. So the mitochondria is part of the machinery of the cell, and the ATP that is produced by the mitochondria can be used now to make more viruses. So when we think of cellular machinery, we should think about the organelles that we've been talking about. And you should definitely go back and review eukaryotic and prokaryotic, plant and animal cells. You should definitely go back and review that. Review the organelles. What is the role of the ribosomes? What's the role of the rough endoplasmic reticulum, the smooth endoplasmic reticulum? Lysosomes, centrioles, etc. You will be taking a final exam, and it's going to cover a lot of information. You need to use this time productively. Well, we've already talked about the capsid. So the capsid allows the virus to bind to the cell and enter. It also protects. So this is protection as well. Now what's going to happen, the cell is now, it has been commandeered by the virus. So the cell is going to transcribe and translate. So this is transcription and translation of the viral genetic information. And now the cellular machinery is directed at creating more viruses. Viruses would have many fewer genes compared to a prokaryotic or a eukaryotic living creature. I mentioned it before. Viruses are very specific. A plant virus will attack only a specific species of plant. The tobacco mosaic virus, that attacks only tobacco plants. Animal viruses will attack only specific types of animals. Bacterial virus or a bacterial phage will attack only bacteria. Now I do want to go back here to this animal virus. Viruses are specific, but, and this is a big but here, capital B-U-T, viruses will mutate and change and therefore have the ability to adapt and evolve. It's a very good example of natural selection when a virus adapts or evolves. What do we mean by that? A virus can adapt and evolve and then actually jump from one animal species to a different animal species. HIV probably jumped from a chimpanzee to a human, and how does it do that? It evolves or it mutates. I mentioned before that mutation is the engine for evolution and natural selection, and mutating viruses will result in viruses that become adapted to new cells, of which now they can become cellular parasites. Now go ahead and copy So, I'll give you a little time here. All right. Let's go through the two different types of viral infections. Again, just listen as we go through, and then I'll give you some time, or you can simply pause the video and you can write down the information. So, two types of viral infections. I've already mentioned this, the lytic and the lysogenic viral infections. Lytic infection. The virus is going to enter the cell. The virus will commandeer the cell, take over the cell, use the cellular machinery to start making copies of itself. Within a fairly short period of time, that cell is going to burst. It's going to lyse. That's why this is called lytic. A lytic infection causes the cell to lyse or explode. New viruses will then be released, and these can go and infect other cells. And we've talked about exponential growth. Viruses will reproduce and cause infections of new cells exponentially. One virus infects a cell, takes over the cell, utilizes cellular machinery to produce more progeny viruses. That cell then dies. And by the way, this is why you're getting sick when you have a viral infection. The parasitic virus, causes cell death. New viruses are released. Let's say one virus infects, it takes over the cell, it produces 10 progeny viruses. The cell lyses, explodes, releases. Now you have 10 viruses going out and infecting 10 healthy cells. Each of these viruses will now reproduce, and each of those 10 cells now produces 10 more viruses. So these 10 cells die. They're each releasing 10 more viruses. It doesn't take long before millions of cells have been infected and the person is getting sicker and sicker. But have no fear. Have no fear. The human immune system is here to start taking those viruses out. There we go. So the lytic infection. The flu is a good example of a lytic virus. Poliovirus is a good example of a lytic virus. So get both of those. The flu is a lytic virus. The poliovirus is a lytic virus. The flu, and I want you to get this as well as what I'm talking about now. So the flu is specific. It's going to attack and inject its DNA into respiratory cells. Polio is going to attack and infect nerve cells. And it's going to be a lytic infection. And again, we're going to have a situation where one virus infects one cell, but that cell is then going to be used to make many more viruses. The cell is going to lyse, and each of those new offspring viruses go out and attack new cells. And this has an exponential effect. I'll say it again. A lysogenic infection. Let's take a look at what happens before we write. Let's take a look at what happens with a lysogenic infection. So now the virus is going to integrate its DNA, remember viruses can have DNA or RNA, into the host cell. We now call that viral component, once it's embedded into the host DNA, we now call that a prophage. So that sneaky little tiny virus has now incorporated itself into the host DNA. So the viral genes now are going to replicate along with the host cell's DNA. The host cell is going about its normal day-to-day cellular business, and it is ignoring the viral DNA. The viral DNA is inactive. So the viral DNA is inactive. At some point in the future, some factor activates that prophage, then it's going to Once activated, it's going to begin viral or virus synthesis. And at that point, we're going to move into the lytic phase, which we just talked about. That lytic phase would be a cell starts reproducing viruses. In short order, the cell's going to lyse or explode, releasing those viruses to infect new healthy cells, and that process continues itself in an exponential fashion. HIV, and I want you to get this, HIV, human immunodeficiency virus. And this causes a syndrome known as AIDS, and we'll be talking more about this soon. HIV is a lysogenic virus. So HIV, lysogenic. I want you to think of a lysogenic virus, think of it as a time bomb. Okay, this is chapter 21. This is lesson 1. We are taking a look at a lytic infection versus a lysogenic infection. I want you to copy this out completely, label it, take a picture, and send it to me. So you're copying this completely, labeling it, and you're sending a picture to me. Make sure it's completely labeled. Let's go through the steps here. So here we have our little spaceship-like bacterial phage here, or phage, and here's our unsuspecting bacterium. So notice the first step here. We are going to attach, so the virus is going to attach and inject the viral DNA into the host cell here. Now notice, we're going in the lytic infection here. So lytic. The viral genes are then going to be transcribed. This is DNA to RNA to protein. Transcription and translation. Important term there. So the viral genes are going to be transcribed by the host. Notice the bacterium makes new viral proteins and nucleic acids. So these are the parts of the virus here. Again, the host has been taken over. So what we have is a terrorist takeover of this bacterial factory here. Notice the proteins and the nucleic acids are going to assemble into new viruses. Next, the viral enzymes are going to cause the bacterial membrane here to lyse. That means break open. And out go these bacterial phages. These new viruses escape and infect other bacterial cells. So notice that the lytic, and this is important, the lytic infection results in cell death fairly soon. Notice in the lysogenic infection, we attach, we inject the viral DNA. Notice the viral DNA is now incorporated into the host DNA as a prophage. So that's an important term there. It's now a prophage. That prophage is a time bomb. This prophage will remain inactive for varying lengths of time. HIV, after infection with HIV, the lag time there is going to be anywhere from 5 to 10 years. So the HIV virus has incorporated into the host, and it's just biding its time. Notice this virus as part of the host DNA. When the host undergoes fission here, which is cell division, notice the prophage just replicates right along with the rest of the DNA. At some point in the future, the prophage can exit the bacterial chromosome and then enter this lytic cycle, resulting in cell death. All right. So again, I want a nice, neat copy, completely labeled. Find this, chapter 21. This is lesson 1. So use your books here. All right. I'm going to sign off just for a moment, and then we're going to be talking about HIV.